I'd like to ask all of our speakers for this afternoon's session to join Dr. Jacob Brown and myself to moderate this open mic session. Perhaps I can lead off with a question comment. This was brought up several times by the various speakers in a particular way, a little bit different for each one, but relates to finding the right balance between something that each one of you had mentioned in some way, of recognizing that on the one hand, all of us like to have good evidence-based medicine to make decisions, and yet with the shrinking pipeline to go forward, perhaps funding mechanisms out there to enhance retasking drugs and utilizing the wealth of data being generated now in the genomics world, it's often inevitable that we look backwards on the existing use of medications right now. And I think of clopidogrel as a particular example, but in not having the ideal world available to us in all times to find the best evidence, the question is, or can you comment on the following as to how high should the bar be set to show proof of concept of value? So here we have a drug for which very large clinical trials have really exposed deltas and outcomes that rival that of drug A versus drug B in head-to-head trials. And I'm thinking about the Timitritin 38 trials, which were not discussed here, that showed a 12 or 8% differential between carriers or non-carriers of CYP2C19, et cetera, for clopidogrel. And then you add to that meta-analyses that have been published subsequent to that. And then you add to that the wealth of data that have a mechanistic rationale behind this. Yet the noise and the sense we get is not enough. We need prospective evidence-based trials. And that seems like a reasonable argument on the one hand, but for those individuals that have a stent placed and their physician or clinician refuses to, and does in fact follow the AHA's guidelines about not getting a genetic test, it falls somewhat flat in appreciating basically who can push that envelope a little bit further, since it's now me involved or a loved one or something like that that's involved. I mean, we don't do prospective trials to test the systems that we have for when a physician or a clinician, he or she decides to take an MRI because they suspect something, which is a very expensive test to do, or perhaps a fasting lipid panel when we know they're not fasting. We don't call those questions and show proof of value of those things. So how high should that bar be set is my question as it relates to that particular example, but you could expand it to others. Anybody can answer that. Comment on it.
I'll start if that's okay. So I think that, when I think of that, because we answer this question a lot, how high should that bar be set? I think there's a couple of corollary questions. One being, how high is the bar set for information that we use in the patient care process in the same way? And so I guess if we really look at pharmacogenetics as a piece of information that informs the care of patients, but not as the only piece of information, then I think it begins to make sense in the context of a serum creatinine and creatinine clearance, these other kind of measurements that we've seen that we also don't have randomized controlled trial, clinical trial data to support. And so I think that putting it into context for the clinician as to what it is, and I also think realizing that this kind of argument, or it's not really an argument, but this discussion and conversation for a randomized controlled trial isn't exclusive to pharmacogenetics. And so, I think looking at other areas where it's not feasible or it's not it's not kind of rational to have that prospective randomized controlled trial data, making the best decision with the evidence that we have, with the best evidence. And so I think having that conversation and acknowledging that, that it isn't something that's present for that, but at the same time saying, is that something, like, how do we feel about that? And I think putting it in that context and saying that it's not the only thing, that we're not advocating you do this for every patient, in every situation, every scenario. And I think keeping it in perspective helps in having that conversation. And I think really promoting appropriate use of the test, and rather than kind of broad-based for every single patient every time. I think that's one of the things we can do as clinicians, is really help other clinicians understand appropriate use of pharmacogenetic testing. So.
Thank you.
So
you want to comment?
I'll comment on that. Personal opinion, obviously. These are my own views, not FDA's. I have big challenges with the prospect of doing a randomized trial of testing strategies for every genetic marker. I think First of all, they're inefficient trials. I mean, they're designed to fail. How can you possibly power a trial where you're only accruing a difference in 5% of the population or 10% of the population? When 70 or 80% of your trial gets the same treatment in both treatment arms, that can't possibly be successful. I understand the importance from a health economics and clinical utility standpoint, but the Abacavir trial is the exception. There's only been a couple of other successful cases of it. So I think we really need to be creative about what kind of evidence and what kind of trials should be conducted. And you could very easily also do a trial of warfarin in sensitive and presumed non-responsive patients only, and then simulate everything else in the middle, and then come up. I mean, so there's a lot of different ways to look at it, and then whatever health system could make the individual determination as to whether or not paying for those 5 or 10% of people would be worthwhile. You know, there's a lot of things like cluster randomized trials, other approaches that I personally haven't seen too often in this field that could be potentially very helpful. And with all the health systems collaborating and working on these things, you know, it might be time to consider those approaches.
Steve?
I think on that question, we have to differentiate the trial or the trial design, randomized trials versus other designs, versus marginal effect. Even if you use other designs and you still only have a very marginal effect, and it's questionable whether that has clinical, it may have statistical significance but not clinical, that's a very different question than saying, do we have to have the ideal trial design? So I think if you use some of these other trial methods and show an effect that's not only statistically but based on clinician input, clinically significant, I think that meets the bar. But if it's only a statistically significant marginal effect, it doesn't meet the bar.
Sure.
Go ahead.
And I'll also say, just from the evidence standpoint, a lot of what we do is based on pharmacogenetics and pharmacodynamics. I mean, basic clinical pharmacology principles, renal function studies, hepatic studies, that are all the basis for all the recommendations we have in drug product labeling are based on pharmacogenetics for the most part. And that's largely, I think, how we view genetics. It's just another intrinsic factor that determines drug response. From the utility side, you know, I also, there's a lot of ways to use this information as well. You know, 5 milligrams of warfarin works because it loads a patient who's a poor metabolizer. The only problem is we don't know that patient's a poor metabolizer and we don't reduce the dose, which is why we bounce around for months after. You know, and there's basic clinical pharmacology that I think goes into, you know, the use of some of these drugs as well as the tests. And when they're appropriate, you know, it may not be that warfarin algorithms should be used to select an initial starting dose for every patient, but that information still tells you when vitamin K intake is more important, perhaps, or how often you should dose adjust.
And I was just going to add, I think we need to also kind of be purposeful and careful on developing our systems and our responses in such a way that we're not just kind of advocating for that payment or that inclusion or that approval of the test within whatever infrastructure that we've talked about, but we're also kind of making sure that we don't forget about the mechanism to respond to the test when it already exists. Because I think we definitely see more of that within our health system of, you know, the question is, now what do I do with this? More so than should I, you know, kind of how do I get it? And so I think it's important that we prepare ourselves for both sides of that and don't get caught up worrying about when, and maybe things are already moving up here and we miss that because we're thinking back here, so.
Go ahead.
I would like to add to the following comments that to me, the notion that if there is validated reproducibility of the predictivity of a genetic marker for response to treatment or toxicity, I would go as far as to say that it would be unnecessary to go to a clinical trial phase. The reason is that the clinical trials as a randomized experiment basically are designed to eliminate confounding. And when you have a genetic factor, it's not confounded by something else. It's fixed for the patients. And therefore, there's no need for randomization in that sense. Furthermore, there's an ethical concern that if you have established that you can reliably predict responsiveness or toxicity, entering into a randomized experiment where you do not use that modality for the conventional non-diagnostic test arm of the study would be unethical, in my opinion, for that arm, for the patients in that arm. And also, I want to mention that we have to be careful as to what is the exact design of this whole process. Are we talking about markers and diagnostic companion tests that are added ex post facto, so there is a marketed drug and we try to create companion diagnostics, or the development of the companion diagnostics is embedded into the clinical trial process in the form of patient-centric or molecular clinical trials? I think these are two different animals that we have to really separate in terms of design.
That's true.
Brian? I have a question about the regulatory role of the FDA as it relates to laboratory-developed tests. So the knee-jerk reaction is that a test needs to be appropriate, quality controlled, valid, and so the knee-jerk reaction is, oh my God, we have to regulate this. But the accusation to the FDA is that they're getting dangerously close to requiring the demonstration of efficacy for drug selection or for diagnostics, and they are getting dangerously close to dictating the practice of medicine. Do you want to comment on where the FDA is I mean, I know that's a debate that's going on as to where it draws the line on what should be regulated and how much should be retained within the art of medical practice.
Yeah, I think Kristen can probably answer that better than I.
That's all you, Mike.
I unfortunately can't comment.
So you can't comment. So there is a discussion about where to draw that line currently in the FDA, correct?
Laboratory developed test regulation is a very intensely interesting area at the moment.
Geez, I waited all that time for that question, and that was my answer. Oh,
You should have asked me last night.
So I have a little bit of change of topic here, and I was glad to hear about, the history of this negative view of genetics. And I wonder if I could get just your thoughts on, as we look to maybe a more standardized approach, if we keep this kind of abnormal, normal type language that is more historical as it relates to pharmacogenomics, are we still going to have that problem or instill it in patients that this is kind of a scary thing versus in pharmacogenomics where it's more of a continuum? You don't have a lot of enzyme, you have a little bit, you have a lot. I mean, we saw here at lunch, if you look at the panel, no one was normal. And patients really get concerned about seeing abnormal, usually in red. I mean, we saw a lot of what was viewed on those things, and they're in red, that's bad, versus just, this is variation, and this just means you don't do well with this drug, or you should go on this drug. And really taking that just negativity away. If you could comment on that.
Let's take that.
One thing, I think the disease marker gene tests that say you will get or you're likely to get this disease, those are pretty likely to happen anyway. With pharmacogenomics, we can say, if we understand your genes better, we can help you get better results from your drugs. Even if we don't understand it, it's not necessarily a scary thing. And the other thing is, we can create the concept we're normally different rather than normal, being good and bad differences. We're all different, but that's normal. That's the usual. Just so we understand, are you a green one or a red one or a blue one? And how does that mean we should use drugs to treat you effectively? How can we use that to help you? It becomes a positive conversation instead of a negative conversation.
And I think it points to something that we kind of see a lot within our Ignite network, which is kind of a conglomeration of a number of very different, diverse, both testing and clinical environments and also interventions. And when we talk about patient education in that group, we see such a broad difference between kind of where we started with genetic counseling and Mendelian risk assessment to low-risk genomic testing or pharmacogenomic testing, and where is a genetic counselor needed? How do we communicate these results? Does it require a sit-down, meeting? Is it something we can tell patients in a letter? There's a lot of unanswered questions, I think, in that area now, and so what I think I've kind of seen coming out is really more of a standardized, kind of helping to develop standardized approaches, whether that's an algorithm where you say, if this, then that, this is where I need to pull in a genetic counselor, this is not. But I think putting that infrastructure in place will help a lot, and then also kind of thinking about the context, like you said, of saying not abnormal versus normal, but kind of, I've heard it described as we all are, have different roads and traffic travels at different, paces on the road. And I think it's especially important when we look at different drugs. If you're a poor metabolizer, it's sometimes a good thing, it's sometimes a bad thing, depending on the drug. And so trying to get all that into a quick explanation isn't always easy.
Hi. So one of the best ways to get a pharmacogenomic test into the payment system is to demonstrate that the cost of hospitalization or some medication has decreased as a factor of the test. To do that, one might use a pharmacoeconomic modeling or say econometrics, but the problem is that there is no such valid databases available where it has been shown that what happens after the pharmacogenomic testing is applied. So the cost of hospitalization or medication. So does FDA maintain a database where it shows that the factors that were involved in the medication and then what was the cost before and after the factors, like the testings were used? Or does any hospital maintain any such database which can be used for this kind of modeling approaches?
We have a number of contracts to do pharmacoepidemiology studies and have rolled genetics into some of them to see if it's a feasible infrastructure to look at drug safety questions. The Sentinel Initiative obviously is the largest scale effort as well. We don't deal with the economics or value questions. We're a safety and efficacy public health regulatory agency. So certainly those questions could be looked at in those systems, but not by us.
Thanks.
Germaine Cornelissen, University of Minnesota. First, I would like to thank every participant and presenters. It was very enlightening. One question, though, the way I see in personalized medicine is the need also to consider timing. More than 50% of our genes are circadian periodic, and we have done quite a few experiments testing different drugs, whether it was for cancer or cardiovascular or asthma, showing that there's a very large circadian stage dependence of both toxicity and efficacy of these drugs. So you mentioned that it may be important to test drugs to specific patient populations, but also it would be very important perhaps to also test these drugs at the right time, because at one time they may not be having any effect, at another time they may be very valuable. The question is, what is your take into including into personalized medicine also circadian rhythms and chronobiology more broadly? Not just space, but also time.
Yes, I mean, certainly you could personalize on any number of factors, and certainly that would be of interest to do. Personally, I mean, I can't take an antibiotic twice a day for 10 days, let alone. No, but I'm saying that tongue in cheek, but I mean, seriously, I think any factor that has a significant impact on drug safety or efficacy obviously should be dealt with very, very carefully. And if it does influence it, certainly timing could be very important. We often, for some biomarkers that are used in clinical trials, try to ensure that they're measured at the appropriate times of day, because they do fluctuate quite a bit, and that can significantly affect how you interpret a clinical study at the end of it. It's very important.
I think someone earlier this morning commented that precision medicine has become a bit diffuse in what does it mean and what does it really represent. And I think we need to be careful to distinguish precision medicine or individualized care as not being just boutique, whatever I want to do for you medicine, or eminence-based medicine, but really evidence-based medicine, that I think there can be a science to individualizing therapy, which is different than providing boutique-y, whatever you want type medicine without the science behind it. And so I think we need to make sure that precision medicine does keep a science base to it and to the decisions we make and the way the resources are used and allocated to deliver that precision medicine.
Go ahead.
Of course, with pharmacogenomics, we're focusing on the way genetic variation affects pharmacology, drugs, their actions, side effects. Years ago, I was originally trained as an enzymologist. I remember very little of that. But what I want to ask is, obviously these genetic variations, I mean, we evolved before there were drugs. What is the actual impact of these genetic variations in the enzymes and all, et cetera, on the body itself, outside of the action of drugs? Do we know anything about that? I mean, is there something internally that these enzymes are acting on that some people evolved faster metabolism that was beneficial to them in some way, or slower metabolism, or some other? I just don't know if there's anything like that. I haven't heard about that.
Yeah, I think CYP3A5 expression evolved in a way that it's segregated in equatorial regions because it has to deal with mineralocorticoid metabolism, I believe, and sodium retention and water. So it got preferentially selected in some populations, but as for the others, I'm not quite sure.
That's a reasonable question to ask, because it does present itself as a curious observation that there are certain populations that are enriched for certain genotypes, CYP2D6 in sub-Saharan Africans, for example, as PMs. And it begs the question, what is the anthropological rationale or reason for that evolving that way, separate and distinct from the issue of just low admixture as a function of physical, geographical kind of limitations. But I don't know of too many other specific examples there that might have a rationale behind them other than what you mentioned, Mike. Dr. Alathas.
I unfortunately don't know any other examples either, but I just want to mention that the model of using the electronic health record coupled with genotyping can help address questions of that nature very directly and very inexpensively. Mm-hmm. Once you have the genotypic information, you can see the links with any number of traits, including non-disease traits that are recorded somewhere in the social or other aspects of the patient's history. So there's the potential for making discoveries of that nature with the infrastructure that's being deployed.
So expanding just a little bit on that discussion that you presented on PHWASs, going the other direction there, and in particular taking note of the fact that in a data-rich environment where you have a lot of potential phenotypes, and perhaps a limited genetic profile on individuals here, the capacity exists right now to have both a data-rich environment for phenotypes and, of course, a complete, exome sequencing kind of database right now. And I see you right now sweating a little bit because you're worried about the capability of the data sets and storage and issues here. But that issue aside there, this speaks to the complexity of all the issues that were brought up earlier about other interactions, simply compliance of medications in terms of interpreting longitudinally, how people respond to medications and discerning from that database, linked to EMR data and so on like that, how all of those real life situations can actually be interpretable with this volumes of data. So at one point when eMERGE grows or other systems grow to kind of incorporate vast numbers of not just data, but people as well in their data sets, I can see that being a very opportune situation to really qualify the value and quantify the value of knowledge of genetics as it relates to drug response, but in a real world setting.
If I can add really quick to your thought about this, once this is established, meaning when we have a very rich database of connections between genotype and a variety of traits, not just disease traits, not disease phenotypes, by linking to other online and digital sources, such as online Twitter feeds, discussion groups, etc., you can expand this network of connecting the information together and see the linkage of genetic information to a whole variety of behaviors and so forth. So the power of amassing these data sets, it can only increase exponentially.
Go ahead, Yusuf. Thanks.
I just have a really quick question, and I hope it's not too long. As we continue doing pharmacogenomic-based research, one of the questions and challenges that we face is returning data to participants. And it's a very controversial, and it creates some sort of discomfort, I guess, for some people and some others. I'm just curious from a FDA standpoint, are we seeing kind of a guideline, or is there a specific recommendation on what to return to patients, what not, what criteria that need to be met in order to do that? So if you just have any comments, that'd be great.
Yeah, so again, my reason for being evasive on the lab-developed test question is because, I'm in the Center for Drugs, so I deal mostly with drugs, and we have a Center for Devices. And they're probably the best points of contact for further insight on that. Being done in a CLIA environment and patients are consented appropriately. I've participated in trials before where I was given that option, and I'm not exactly sure what the answer is at the moment. But
all right, I think we've got time.
If you email me, I can get some further information for you.
We've got time for one more question.
So this is potentially a naive question, but I was thinking a lot of the discussion has focused on cost of, or who's going to pay for genetic testing, and that's one of the seems to be one of the barriers. And given that the cost of the kind of testing that we're talking about is what, between $200 and $500? Is that about right? Device that I have in my hand costs $650. And I know that a lot of people basically buy one every year, sometimes twice a year. So I'm just wondering how much of this discourse should be focusing on consumers, and maybe we should be selling this to the consumers, to the patients, that they would see the value in doing this kind of testing and holding on to the data and presenting it, and then educating the physicians on how to interpret these data, so that we sort of don't get bogged down in this discussion of how much it's going to cost, because it doesn't seem to cost that much.
Yeah.
Is that a naive question, or is that?
Oh, go ahead.
Thank you. So if you recall this, I think the last slide that I saw about the cost distribution, it had a component that was assay component. And it's a very small component, actually, to the total cost of pharmacogenomics and pharmacogenetics. Probably with today's cost numbers, about 5%. So you're right, it's inexpensive to assay things, but there are other components in the value chain of pharmacogenomics that are expensive, at least with today's economics.
You're assuming that it's a cost that you incur once and it's used over time. The way it works in the healthcare system, I bill you for one little SNP test. I get another SNP test from another provider, another one from another, instead of doing one study and then having that archived so I can access it. So part of it is how we manage and utilize and monetize these techniques in the marketplace. I think people are using it in a way that they want to divide it and sell you as many little SNPs as they can instead of one for $200. So part of it depends on how we present it to the market too.
But I think we're seeing more and more of that, and that was one of the kind of thought processes behind when we think about, is a randomized controlled trial needed to justify testing, is what do you do when the patient comes to you with a test? Think about kind of that. And we see from a pharmacy perspective, I've seen more and more models emerging. And in this area, this is not a model that we're using at UF, but a number of kind of it's not really direct to consumer. Like the patient is referred for a consult to a pharmacist-run pharmacogenetics clinic. And there's a pre-test and a post-test visit, so that are separated by about 4 weeks. And I've seen this in a couple of different, more in kind of under an MTM type umbrella of service model in an outpatient area. And the people that I've talked to that are doing that are talking in that first kind of pre-test counseling visit, the test, here's the cost of the test. We're happy to submit it to your insurance company. If they don't, cover this in your scenario, this is the amount that you would need to pay. Are you willing to pay that? And I've seen some early data, and again, this is really not, this is kind of talking to different people, but the majority of people, vast majority, kind of over 80%, they're like, okay, that's fine. They're perfectly fine with paying for that. And then I think, and that's usually in a preemptive panel-based model, and I think the key there is making sure that they understand that that's their data, they need to keep it with them, and you can help them interpret that and apply it. And but I think, to me, that's an existing model within pharmacy practice that that can be extended to pharmacogenomics thoughtfully and appropriately and purposefully. So.
I think we'll have to end it there. I'd like to express my appreciation for both my co-monitor, Jacob Brown, and the panel here as well, Drs. Kirsten Weitzel, Dr. Konstantin Aliferis, Steve Schondelmeyer, and Drs. Michael Pakanowski and others. Thank you all very much for this panel discussion, as well as your presentations this afternoon. And as a final note, I'll just make my plea to express my appreciation for all of those that organized this as well, and the vision that Dean Speedy had and Pam Jacobson had, and the organizing panel for this group to put together such an interesting day's event. So my appreciation to all. I'll let Pam close things out, and thank you all once again. Have a good day.
I will be brief. We're nearly at 5 o'clock. Thank you all so much for participating today. I hope it's been an enjoyable day and you all have new insights into personalized medicine and that your educational needs have been met. I also hope that you are now further enlightened and about the emerging field of personalized medicine and the potential health benefits. And you've all been motivated now to go back to your clinical sites, hospitals, clinics, and try to incorporate pharmacogenetic testing into your sites. And again, I want to thank Dr. Speedy, just as Dr. Straka has, for her vision in pharmacogenomics, the College of Pharmacy, the Department of Experimental and Clinical Pharmacology, and of course all of our sponsors who are here today, which would not have been possible without their support. So in closing, have a good evening, everyone, and hopefully we'll see you at the next Precision Medicine Conference. Thank you so much for attending.